Hello, this video is a continuation from the last video when we got cut off, so we're going to continue about genetics. Where we left off, we were talking about DNA replication. So DNA replication is the process of making a new DNA. Where I left off is this term called semi-conservative, meaning that we keep some of the original DNA. So if this is the original DNA, we separate it, and then we build the corresponding bases and the corresponding backbone to the other side. If it was fully conservative, basically we wouldn't break this apart. It would just be a copy of this, like we'd run it through a copy machine, and this would never be interrupted. So when we say semi-conservative, what we're saying is that we have to break it apart and build the new pieces. So we are keeping it, like half of it, and then making the new half. These are diagrams of the what we're talking about in DNA replication. So if this is DNA, we untwist it. We have different enzymes here that are working on either separating it or building the new piece. And we'll talk about what those are in these next few slides. So first off, there is one of the enzymes is called the helicase. And the helicase, what it does is it unwinds the DNA. Like it starts to untwist it and unzip this DNA. And that's called it's at the origins of replication, like where replication is going to start. Then the initiation proteins, they form a replication bubble. Like they literally create this zone where it's going to start making the new DNA. Then we have primase. I don't know if you've ever done painting before, but you always set like an initial layer called a primer. Same thing, we're going to put down an RNA primer, like basically like saying this is where we're going to start replication. Then there's an enzyme. It's called DNA polymerase 3. And what it does is it adds complementary bases to the leading strand. So remember when we unzip the DNA, and I'll show you a picture, part of it's building towards the opening of the zipper, and part of it's building away from the zipper. The one that's building towards the zipper is called the leading strand. The one that's called the opposite side, that's building away from the zipper, that's called the lagging strand. And if you remember from the sample test we did, it builds it in pieces because it's building away from the zipper. And those are called Okazaki fragments. And at the very end, DNA polymerase 1, another enzyme, takes away that primer that we started with at the beginning and we make it with DNA. And then DNA ligase is basically another enzyme. And remember, we build in pieces. So this one seals all the fragments and puts it all into one piece. So here, basically more detail of each of those steps. So remember, number one, we talked about helicase. So helicase unwinds it. So if this is the original DNA right here, then it unzips and creates these bubbles that we were talking about. So here's a bubble, here's a bubble, here's a bubble. Okay. If you want a molecular picture of what it looks like, you can see these little bubbles of the DNA just kind of opening up. Remember we talked about primase. So if this is my DNA, here's where it's being unzipped. Yeah. Here's that helicase, the one that's doing the unzipping. See how it's going towards this zipper and unzipping it. And the primase, remember it adds the primer, it's building an RNA, basically saying this is where we're going to start the replication. And then there's this topoisomerase, not so important to know, might be on the biotest, I don't know. But it basically kind of holds this into place, kind of like a clamp when this helicase is unzipping. And then we talked about DNA polymerase 3. It builds from 5 to 3. So remember we talked about how DNA is labeled. One side has the 5 carbon, and at the other end it has a 3 carbon. So it always builds towards in the direction 5 to 3. Okay, and it's on the leading strand. That's what DNA polymerase 3 does. So here's an example of what we're talking about. So here in this direction, you see how it's 5 to 3 on DNA? What this is saying is that everything builds from 5 to 3. So if this is 5 to 3, this dark blue line right here, the new one that I'm building has to be built this way because it has to build from 5 to 3. So these are complementary, and these would be complementary. So after I make my primer, and here's another sliding clamp, my DNA polymerase 3, which is this piece right here, is building the second part of the DNA in that direction towards the zipper. And that's called the leading strand. And later on down the line, this is what it looks like even further. Again, it's building DNA down from 5 to 3 into the zipper, called the leading strand. But the opposite side is called the lagging strand. It also, remember, you can only build from 5 to 3, but if you're building away from the zipper, you have to wait till it unzips a little bit and then build. Then unzip a little bit, then build. And that's why they call that the lagging strand. So here, they call this an Okazaki fragment. Again, they're short segments that they build, and then they have to wait for it to unzip, and then they build again, wait for it to unzip, and build again. So here it's hard to see in this picture, or hard to understand what's going on here. But again, it builds from 5 to 3. So here you see 1, here you see 2. It keeps building in pieces. And then the DNA ligase, which is... see how these are in pieces? I have red, blue, red, blue. I have red, blue, red, blue. This DNA ligase seals the fragments, so I don't have those red primers, those RNA primers anymore. It seals the fragments together. And here's a nice table of all the DNA enzymes and proteins, what they all do. We talked about helicase, which unzips. Topoisomerase, again, it's kind of a clamp. It relieves overwinding, so it's at the end and clamps it. Talked about primase, making the RNA primer. DNA polymerase 3, which builds on the leading strand. Polymerase 1, which removes the RNA primer. And then DNA ligase, which we said when we make in fragments, it seals all the fragments up to make one nice long strand. So here's a big picture of everything that we're talking about. Here's the leading strand that goes into the zipper. It builds in one nice, clean, continuous strand. And on the other side, the lagging strand, it builds in pieces here and here and here and here. So you can see the red being these RNA primers, but it has to build in pieces. And then the ligase, which is right here, seals all the pieces, so it's just one strand. And then at the end of it, they have to proofread, because you just built a whole new DNA. You need to make sure that it's correct. So the DNA polymerases, as they add the bases, they also proofread to make sure that everything is being copied correctly. If there's a mismatch, there's something that repairs it, and special enzymes do that. One of the kinds of repairs is called a nucleotide excision repair. Excision means to cut. So these enzymes called nucleases, they cut the wrong or damaged DNA, and then the polymerase and ligase, they fill in the gaps, just like we did in the lagging strand. So here's a picture of what the excision repair looks like. First of all, the errors, there's about like maybe 1 in 100,000 nucleotides that are incorrect. And in a complete DNA, like of the 10 billion nucleotides, maybe only 1 is incorrect. Okay, so I mean, like, the margin of error is very small. And this is what happens. So here's the thing that's wrong. So the nucleus comes in and takes these pieces out. It does an incision. Then the DNA polymerase builds the new one, and then the ligase again seals them together. So here's the problem at the 5-carbon end. So the DNA polymerase only adds nucleotides to the 3' end. So there's no way to complete the 5' ends of daughter strands. So over many replications, DNA strands will go shorter and shorter and shorter. So basically, when we remove the primers and do all this stuff, I get like basically I'm a little short. So basically, my new DNA is always a little bit shorter than the original DNA. So what they're saying the problem is that as I go more duplication and more duplication, the molecules get shorter and shorter because there's no way to finish this end. And that's because of the, as you can see here, it's because of that zipper part, the unzipper part. The very, very, very end of it is always going to be empty. Some other things that you want to know about telomeres. They're repeated units of short nucleotide sequences like TTA, GGGG. So at the end of DNA, they just keep repeating this nucleotide sequence. And what that does is it makes a cap at the end of DNA and postpones erosion of genes. So basically, if at the end of remember I told you it always gets shorter and shorter and shorter, or in case there's damages at the ends, they basically repeat these things, TTA, GGG, that are dummy nucleotide sequences. So if anything happens, I just got rid of or ruined parts that I didn't really even need. And the part that does add these TTA, GGG, that enzyme is called telomerase, which adds to those telomeres. So a new topic, so that's all DNA replication. So the central dogma of biology is you have DNA, which makes RNA, and RNA is what codes and makes the protein. And the proteins are what we need. It makes enzymes, it makes our bodies. So that's how genetic information flows. The central dogma is DNA to RNA to protein. Transcription is how you go from DNA to RNA, and translation is how you read RNA to make the protein. And this all happens at the ribosome. Okay? And gene expression is basically which pieces, the process by which DNA directs the synthesis of protein. So when we say like the genes are expressed, we're basically saying which part of DNA is copied, which is then read and creates that protein. That's what I mean when a gene is expressed. So a big picture of what's going on here. This is in a bacterial cell. The DNA you make a copy called mRNA, which then goes into the ribosome and makes a polypeptide or a protein. Because it's a bacterial cell, they don't have a nucleus, so that's all happens inside the cell. Whereas in a eukaryotic cell, we have a nucleus. So this all happens inside this purple thing. This is the nucleus. We do transcription, which means we take DNA and make a copy of it and call it mRNA. The mRNA then leaves like a ribbon, and then it goes to a ribosome and it builds the protein that way. DNA and RNA, you should know the difference already. DNA is double-stranded, it has deoxyribose and it has T. RNA is single-stranded and it has U, and it has ribose instead of deoxyribose. And then one gene is equivalent to one polypeptide. So when we make a polypeptide, we're making a gene, basically. So here are all the RNAs. There's 7 RNAs you need to know, and they're not that difficult as you think. Pre-mRNA, M stands for messenger RNA. So the messenger RNA is basically the messenger from DNA to the protein. It just is transcribed. It just took a copy of DNA, and it hasn't been edited yet. Okay. mRNA is the official messenger RNA, and what it does is that after it takes a copy of DNA, it kind of edits it so it carries the correct code. It also it's gonna have a cap and a tail, so it's like an edit. It's like the final draft, and this is like the rough draft. Pre-mRNA and mRNA. tRNA, and again we'll talk about this when we have pictures. It's short for transfer RNA, and it basically transfers an amino acid or like a bead, a piece of the protein, and it transfers it. Transfer RNA. rRNA is ribosomal RNA. So when we talk about where we're making the protein, it happens in the ribosome. rRNA, ribosome RNA. sRNA, it's a small nuclear RNA. It's part of a spliceosome, and we'll talk about that later. SERP RNA, so it's a signal recognition particle. And then there's RNAi, interference RNA. We'll talk about, again, all of these in the next few slides. So let's start from the beginning of transcription. So here's my DNA, and what happens is that I separate the DNA and I make the copy or the complementary bases. So you see here A, C, C, A. When I transcribe it, it becomes U, G, G, because the partner of A is U, partner of C is G, and partner of C is G, A is U. So this red right here is the partner of this top piece right here. And then in translation, basically these letters are in groups of 3, and they call them codons, and each group of 3 translates, this is translation, into an amino acid or a bead, so to speak. And all these beads hooked together make up a protein. So this is a nice table that you need to know how to read, okay? And I can tell you how to read this very quickly. So basically, this is the first base, this is the second base, and this is the third base. So if I want to figure out what codon, again, there are 3 letters, what amino acid I'm going to get from it, you just give me the letters and I'll tell you what you make. So say, for example, you give me a letters AUU, okay? So here's A, so it's this row, U, so I'm here, and then U again. So AUU is right here. This tells me the amino acid I need is called ILE. This is what we did in class. I don't know if you remember doing this activity, but we put all these all over the floor, and then I had you guys find the amino acids and tape them together. We have redundancy, which is basically we have 1+ codons of code for each of the 20 AAs. And also we have a reading frame, which means, remember, we read in only in 3s. So this code is universal, so no matter what organism you are, they all use this same code. So in this particular case, when we're talking about transcription, here's basically what's going on. Transcription is the stretch of DNA that codes for a polypeptide. Again, a polypeptide is a fancy word for a protein. Okay, so we start off with RNA polymerase. So last time we just talked about DNA polymerase. RNA polymerase separates the DNA strands and basically, again, makes the partner of DNA. It also goes from 5 to 3, and when it makes the new mRNA, we have U instead of T. It starts at the promoter, which is the start of the gene, and then stops at the terminator, which is the end of the gene. Okay. So what we're going to do is what they call a couple of steps. First is initiation. Okay. Initiation, we start at what they call a TATA box. I know it sounds funny, but TATA. Tata box is a DNA sequence that's upstream from the promoter, and basically it's where we start. Wherever the Tata box is, it helps us to know where we're going to start. Then the transcription factors, like these little enzymes, they recognize the Tata box before RNA polymerase can bind to the DNA promoter. So here's step 1. Okay, I've got a promoter. I got 5 to 3. Here's a Tata box. Here's a start point. The transcription factors recognize the Tata box over here. And now here's my transcription initiation complex. This big old thing is RNA polymerase. And it's able to start at the start point because we recognize where the Tata box is. Okay. Then we have elongation. Elongation means to make longer. So what we're talking about is we're making our mRNA longer. So this RNA polymerase, this giant thing we talked about, is as it goes on, is separating the strands and making the complementary bases to the DNA. So you see here TAG, so now I'm making AUC, and it comes out, the newly made RNA comes out this way. And as it goes and unwinds it, as it moves on, it winds it back. So the RNA polymerase adds RNA nucleotide to the three carbon end of the growing chain. So as it untwists, again, as I was telling you, as it goes across, it untwists it, and as it moves across, this becomes wound again. So it's hard to visualize, but this little white thing goes across the DNA, untwisting and retwisting, and while it's doing that, it's making this ribbon of mRNA. And then termination. So we talked about there's a terminator sequence, so we start with the Tata box. So as I go all the way, and at the end, there's a terminator sequence, which means the polymerases detach. And then we now have our pre-mRNA. Okay. For prokaryotes, this is ready to use. You don't need to have pre-mRNA because it doesn't travel. The reason why we have to edit this is because it's going to travel throughout the cell. So what are the edits you're talking about? So we built this. We have this pre-mRNA. What do we edit? We basically add what they call a cap and a tail. So this red is my mRNA. It's my pre-mRNA. My real mRNA is I add something to the front and to the back. The front gets what they call a 5-carbon cap. So it's basically a modified guanine or G and a whole bunch of phosphate. So it's the cap of my mRNA. And at the end, I have what they call a poly-A tail, where I add like basically a whole bunch of A's. 50 to 520 A's are added to my mRNA. And the reason why we do that is because this is going to leave the nucleus and go to the ribosome. And if it's going to travel, we're scared like something's going to happen to it, might get damaged, stuff like that. So what's going to happen is that I attach this cap and tail as kind of like protection, and it's allowed to travel throughout the cell. We also are going to splice some things. So some things in this RNA we need and some of them we don't need. Okay. Some of the code has what they call introns. They're non-coding. We don't need the introns. Okay. So for example, look at this mRNA. Do you see how it's red and peach? Well, these red ones, the ones that we actually need, are called exons. They code for the gene. These ones are important, but they're not important to what we want to make today. So these introns, I cut them out. Where I cut them out, then all the reds are spliced together. So this is a true mRNA. This is pre-mRNA. It's all long and has things we don't need. And when we cut it and paste it together, I have mRNA. So splicing, you take out the introns and you keep the exons. And then we do some more splicing. Okay. These are called SNRPs, SNRNPs, small nuclear ribonucleic proteins, SNRPs. So SNRPs, they're the one that recognize what needs to be cut. They recognize the splice signs. They join with other proteins to form a spliceosome. So here's a SNRP, and they join other proteins, and they make this big old thing called a spliceosome. And the spliceosome, as you can see, it kind of like pushes this together and cuts out this whole peach thing and puts the exons together. So a SNRP is a protein and joins together to make a spliceosome, and then it splices it out, and that's how you cut out, that's how you do splicing. So somebody's saying, like, why do you have introns? Like, why do you have pieces on the RNA that you don't even really need? Well, remember, there are many things that you may want to make and may not want to make, and you may need it or some don't. This is how you regulate gene activity, okay? So alternative RNA splicing is that you could produce different combinations of exons just based on what you decide to cut and not cut. It's kind of like making a recipe. Like every day, if you were to cook every day, you have all these ingredients, but there's some days where you don't need all the ingredients. You just take out what you don't need. So That's why we have introns. So that was transcription, where you take DNA, you make pre-mRNA, you add a cap, you add a tail, and then you cut out all the introns. Then you have mRNA. This mRNA, the messenger, is now going to go to a ribosome, which is the site of translation. And tRNA is going to read my mRNA, as you can see in this picture, and deliver the amino acid that is needed. So tRNA stands for transfer RNA. And what happens is that this is what it looks like two-dimensionally. Three-dimensionally, it looks like this. Basically, I have an anticodon, the match, and an amino acid attached to the top of it. Okay. The base pairing rules between third basic codon and anticodon are not as strict, so that's why they call this a wobble. Okay. So this is what it looks like. So say for example, actually we'll skip this one. Here's my ribosome. Okay, this blue and this yellow right here. We have 3 sites, A, P, and E. And I'll tell you what those are. And this rRNA, this ribosome is made up of rRNA. Okay, and we have 3 sites inside, E, P, and A. Okay, A is where it holds the part, and it's where things are added. Okay, so it's the amino acyl tRNA binding site. It's where things add. The P site holds the polypeptide chain, so P, and then E is exit. Okay, so what happens here is that, I guess, again, it's hard to explain here, but look at this mRNA down here, right? The tRNA that matches the codon, that has the correct anticodon, attaches. It's at the A site. It adds. Okay, then it moves over to the P site, and the P site takes that bead and pastes all those beads together. And at the E site, the tRNA leaves and goes away. That's how it works. So again, in translation, we have similar steps. We have initiation, where again, the tRNA goes to the A site and it has this purple thing attached to the top of it. So here you go. Then it moves on to the P site, and again, adds the beads together. And again, the E site is where it leaves. Here's elongation. So again, it sits on the A site, attaches, leaves on the E site. So here's a picture. It was on the A, then it moves to the P. In step 2, a new tRNA moves to the A. At P site, these new purples all attach together. So see how it snaps together? And then this P moves over to the E and it leaves. The A now moves to the P and now has all of the beads at the top. That's elongation. And then termination. In this mRNA, there's something called a stop codon. So when it gets to the point where it's like stop, then you see this yellow thing basically stops the whole process. All these beads remove and leave. It becomes a protein. And then this ribosomal unit, this rRNA, it looks kind of like a sandwich. It breaks off apart as well. Polyribosomes, they are a single mRNA that can be translated by several ribosomes at the same time. So here you go. Here's a picture of a polyribosome. It's a ribosome that works with like other ribosomes. All right, and then the last piece, I guess you could say the last piece of this presentation, we're talking about proteins and protein folding. This is what Mr. Xu went over with you guys during the year. So during synthesis, polypeptide chains, they coil and they fold spontaneously. Okay, so protein, long beads, and it's kind of like headphones, like they just coil and fold and tangle like spontaneously. So what you have is something called chaperonin. Chaperonin is a protein that makes it fold correctly. So it's kind of like I forgot what Mr. Xu called it, but it's like a machine that you stick like something tangled. Like say for example, you have tangled headphones, you stick it in the machine, you close the cap, it moves, and you open it, and then all of a sudden it's untangled. That's exactly what this is doing. That's what a chaperonin does. So here's my chaperonin. Here's my coiled and folded polypeptide. It sticks in, the cap attaches, and then when the cap comes off, the protein is folded correctly. Other thing you need to know, cellular zip codes. The signal peptide, which is 20 amino acids at the leading end of the polypeptide, determine the destination. So remember, these are a whole chain of beads, right? Well, the 20 amino acids at the leading end determine where you're going. So that's called the signal peptide. So think of, for example, I have a ton of beads. The first 20 beads basically tell the protein where it's supposed to go. And then the SERPs, the SRP, the signal recognition particle, brings the ribosome to the endoplasmic reticulum. Okay. So remember, the central dogma of bio, DNA. is transcribed to make RNA. RNA goes to mRNA, which goes to a ribosome, which makes a protein. That's the central dogma. But there are some things that make it not exactly what you think you want. And these are called mutations. Mutations are changes in the genetic material of a cell. We could have large-scale mutations, which means like it affects a whole bunch of the bases, and they always cause a disorder or death. This could be because of nondisjunction, translocation, inversions, duplications, large deletions when I'm doing the transcription. But more commonly are point mutations, where when you're making a copy or you're making the mRNA, you made a small mistake. Okay, for example, they are what they call point mutations, like one base pair is messed up. They could be called missense, which means, like, say for example, it's supposed to be A, but now it's G. All the beads are exactly the same except for that one bead that was whose letter was changed. Or it could be nonsense, which means instead of changing that one letter and it becomes a new bead, what happens is that it changes the letter and becomes the stop codon. So now it's nonsense. It doesn't make any sense. You're supposed to have a bead there, but now it's the stop codon. There's also another mutation called the frame shift mutation. So basically they read it incorrectly. So it's like you're supposed to read it in threes, right? So what if you start at the wrong place? So say for example you have AG, 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 a whole row of AG, AG. So you read AGA, then you read GAG. So basically, long story short, you're supposed to read it like that. But if I start reading at the wrong letter, then all of my threes, all my codons are all wrong because I shifted over too much. Here's another picture of what I'm talking about. So for example, here's that nonsense I was talking about. So this is supposed to be an A. This is A instead of T, which means this becomes U instead of A, which means this becomes a stop codon instead of another purple. This is a missense. We talked about this. Say, for example, this becomes a T instead of a C, which means this is A, which means this becomes A instead of G, which means I get the wrong purple bead that I was supposed to get. Okay. And then there's also what they call silent. Silent meaning that I changed the letter, but I still get the exact same bead I was supposed to get. Okay. And then what I was talking about lastly, this insertion and deletion, I start reading at the wrong start point. So this one I have an extra A, which means I have an extra U in my RNA and I get to a stop. Or this one I'm missing something and all of a sudden I get all the wrong beads from that point on. Okay. And this one is no frame shift, but one amino acid is missing. So say for example, TTC is missing, which means AAG is missing, which means all the beads aren't messed up, but just that one is missing. So what are some of the things that happen because of these mutations? One of the most common ones you'll see is sickle-cell disease. Sickle cells, I told you that your red blood cells are supposed to look like these nice jelly donuts, but instead they're sharp and look like sickles like this. So what happens is you have pain, you have frequent infections, organ damage. That's what's wrong when you have sickle cells instead of jelly donut red blood cells. This is called a genetic effect. 5% of humans have sickle cell disease, and because it's genetic, those that are in Africa, there's a 25% chance, or you have 25% of the people in Africa have sickle cell disease. It's a point mutation, so this is what it's normally supposed to look like. So here's my hemoglobin DNA, and it copies like this. This is CTT, so this will be GAA, and then GAA codes for GLU, which is a normal hemoglobin. But in the point mutation, instead of CTT, it accidentally mutated and becomes an A. This A becomes a T. This T now codes for U, and instead of being GAA, this is now GUA. This codes for a different amino acid, valium or val, and it makes the whole shape of the protein sharp instead of a jelly donut, just by one little minor mistake. So this is what it looks like three-dimensionally, my nice, awesome red blood cell that's a jelly donut, and this is what it looks like because it's not like that. It's sharp, it has a different shape. Okay, so I guess this is a video. Oh, never mind. But it's a video, but basically it's showing you that it's really sharp at the ends. When you talk about how they look like in your bloodstream, this is why the red blood cells are shaped like this, because they could move and flow freely and squeeze together and go into tight spaces. But if they're sickle cell, they could glob up. You see here how like it creates a blockage, and also like it tends to like stick. So they stick to each other, they stick to the ends. And anyway, it's not very good. And then long story short, just a bit of a review. Remember, in transcription and translation, it all happens in the cell for bacteria or for prokaryotes. And eukaryotes, mRNA has to have a cap and a tail when it leaves. Prokaryotes and eukaryotes, the difference between them: prokaryotes have transcription and translation in the cytoplasm, DNA and RNA in the cytoplasm. The RNA polymerase binds directly to the promoter. Transcription makes mRNA, there's no pre-mRNA, and then there's no introns. So this picture is a nice summary of everything we just talked about in the last slide of this presentation. You have DNA, you have this RNA polymerase, this giant thing that unwinds and rewinds to create mRNA. It gets processed. I cut out all the introns. I get a poly A tail and a cap. It leaves to go to this rRNA or this ribosome. I have tRNA that bring the beads that sit in the A, P, and E sites. These purples, they attach, and the purples, once they're all attached and hits a stop codon, makes a protein, which makes you. So that's how you make a protein. That's the central dogma of bio. And that's, again, review for everything that we had talked about earlier this year. So I hope you understand it. Hope you get it. Hope it's a nice, good review for you all. And I'll see you all in the next review video. Take care.